This week we're going to talk about the nervous tissue, and this is just an introduction to the nervous system in general, which we'll be talking about for the next couple of weeks. So we can divide the nervous system into two major components, the central nervous system and the peripheral nervous system. The central nervous system is composed of the brain and the spinal cord, and the peripheral nervous system is composed of everything else, all of your spinal nerves. So the peripheral nervous system can then be split into the somatic nervous system and the autonomic nervous system. Somatic meaning body, so this is more voluntary. Autonomic sounds like automatic, so these are more involuntary processes. The autonomic nervous system can then be split into the sympathetic and the parasympathetic nervous systems. Sympathetic controls your fight or flight responses, and parasympathetic controls your rest and digest responses. Neurons have a long lifespan, so they have no need to regenerate that often. They have a very high metabolic rate, so they require a lot of mitochondria, and they lack centrioles, so it's not an easy thing for them to do to undergo mitosis and divide. So let's talk about the structure of neurons. Neurons are the functional units of the nervous system. These are the cells. Neurons have a body, or a soma. Generally, they have some types of dendrites on them, which are these little branches that help to sense stimuli. Neurons undergo action potentials, so they propagate electrical signals down an axon. The start of that axon is known as the axon hillock. In action potentials, they will travel down the axon into the axon terminals, which will be communicating with another neuron. So the communication from neuron to neuron is called a synapse. These are constantly being made when you're learning things. The neuron prior to the synapse is known as the presynaptic cell, and after the synapse is known as the postsynaptic cell. Nutrients can also be transported along the axon from the body to the end of the axon terminal is known as anterograde transport, and from the axon terminal back to the body is known as retrograde transport. Neurons can look different from the one we looked at also. So the one that we looked at is known as a multipolar neuron. We have a normal cell body, dendrites, and axon terminals. But neurons can also be unipolar, where there's a cell body in the middle, an axon jetting out from either side, dendrites on one side, and axon terminals on the other. There are also bipolar neurons, where there's a cell body with one long dendrite, one long axon, and axon terminals at the end. Or maybe they don't have axons at all. Maybe they're just a giant cell body as an anaxonic neuron. Or they have dendrites, but no axons. So it depends on the part of the nervous system that these are in, on what type of structure they will have to best fit their needs. We have three main classifications of neurons: sensory or afferent neurons, motor or efferent neurons, and interneurons. Sensory neurons can be somatic or visceral, somatic meaning part of the body or skeletal system, visceral meaning part of the organs. Interoceptors monitor your organs, exteroceptors monitor your environment, and proprioceptors monitor your position of your body and the movement of your body. Motor neurons can also be somatic or visceral, and they can be preganglionic or postganglionic, meaning before a ganglion or after a ganglion. Interneurons facilitate integration between the sensory and motor neurons, and these are found more in more complex responses. Neuroglia are the supportive framework for nervous tissue, so these are helper cells, and these are way more common than neurons. They're actually far outnumber them. Our main types are astrocytes, ependymal cells, oligodendrocytes, microglia, and Schwann cells. And of these five, the first four are found in the central nervous system, and the Schwann cells are found in the peripheral nervous system. Astrocytes are called so because they are star-shaped. They maintain the blood-brain barrier, so they help to protect our brain from any drugs or chemicals that shouldn't be coming into our brain at all. They help to create a 3D framework for the central nervous system. They repair any damaged nervous tissues, and they guide neuron development. They also control the interstitial environment. Ependymal cells produce and monitor cerebrospinal fluid, which helps to act as a kind of cushion in our brain and spinal cord. Oligodendrocytes produce myelin sheaths that surround our axons in the central nervous system. So with myelinated axons, myelinated nerves, we have a much faster response with the action potentials. So most of our nerves are myelinated because they require fast responses. Our unmyelinated axons don't have myelin. Myelin acts as an insulation to our axons. So it speeds up action potentials. This is why animals like koala bears and sloths are so slow, because they tend to have way less myelinated nerves than us. So in the central nervous system, when we have a lot of myelinated axons, that's called white matter. And the places where we have unmyelinated axons are called gray matter. Microglia are the least numerous of the neuroglial cells and are the smallest, which is why they're called microglia. They also act as the garbage disposal of the central nervous system, cleaning up any debris or damaged cells as they go along. Schwann cells are also known as neurolemmacytes, and they act as the same function. They have the same function as oligodendrocytes, only in the PNS, so they're going to form myelin sheaths. Prolonged pressure on a neuron or nerve can lead to prolonged damage. A short amount of time of pressure on a nerve is not necessarily a bad thing. This will result likely in you feeling like your arm went to sleep. But once you relieve pressure from that neuron or nerve, it's able to regain its sensation. But if that happens over a long period of time, like a crush injury, that can result in damage. When damage occurs to a neuron or nerve, what happens is Wallerian degeneration, where the distal point of that axon will degenerate and any debris will be cleaned up. This is way more likely to happen in the peripheral nervous system than the central nervous system. Central nervous system is much more protected. Your peripheral nerves are lying within your tissues that can more easily be compressed. We've talked about resting membrane potentials, graded potentials, and action potentials already. Remember that action potentials are not graded potentials because they affect the entire plasma membrane rather than just a local piece of the plasma membrane. So we've talked about depolarization, repolarization, hyperpolarization, and refractory periods already. So you can refer back to the lecture from last week if you need a refresher on that. But there's also a difference between different types of refractory periods. We have absolute and relative. So remember that refractory periods is the time between one action potential and another. An absolute refractory period means that there will absolutely not be another action potential unless another stimulus is produced. A relative refractory period is basically there's a time limit between action potentials, and another can be propagated once that time limit has gone by. So action potentials are also known as nerve impulses. They're not graded potentials. Like we said, they're propagated. They're all or none. So they have to meet a threshold in order to occur. These impulses can be propagated down an axon via continuous or saltatory propagation. Continuous propagation happens in unmyelinated axons, and saltatory happens in myelinated axons. So between the myelin sheaths on the neuron, there are these little nodes called the nodes of Ranvier. And the nodes are where the action potential will hop. It actually hops between them because the myelin sheaths act as insulation. There cannot be electrical activity at the myelin sheaths. It has to occur between the nodes. So the hopping of this action potential between the nodes of Ranvier is known as saltatory propagation. Continuous happens in unmyelinated axons because there are no myelin sheaths to interrupt the impulse, so it continues just straight down the axon. Synapses, as we've said before, are the connections between two neurons. There can be a presynaptic neuron before the synapse and a postsynaptic neuron after the synapse. And there are two main types of synapses, chemical and electrical. With electrical synapses, there is a direct physical contact between cells. This propagates action potentials really quickly. This occurs in vestibular nuclei, the eye, and peripheral nervous system ganglia. Chemical synapses are the most abundant type of synapse. The neurons in this case send chemical signals. There's a synaptic cleft between cells, so there's not a direct connection. This can occur also between neurons and muscles at neuromuscular junctions and neuroendocrine junctions between neurons and endocrine glands. The chemicals that are released by the synapse are typically known as neurotransmitters. Cholinergic synapses release specific neurotransmitters known as acetylcholine, and these are typically released at neuromuscular junctions, synapses in the central nervous system, neuron-neuron synapses in the peripheral nervous system, and neuroendocrine junctions. There can also be a delay of the arrival of the action potential and the effect on the postsynaptic membrane. It could be pretty slow, which is why reflexes are really important. It's kind of like a shortcut. If we had to wait for our synapses to kick in, it might take a little bit of time to withdraw your hand from a hot stove. That's why we have things like reflexes in place. Synapses can also fatigue. The response of the synapse can weaken because resynthesis and transport mechanisms may not keep up with the demand of neurotransmitters. They can fatigue just like our muscles can, so it may take some time for them to revamp. Neurotransmitters can be excitatory or inhibitory, where they can propagate signals or inhibit them. The effect on the postsynaptic membrane will depend on the properties of the receptor on that neuron. So we're going to talk about a couple of types of neurotransmitters. One is norepinephrine. Its specific types of receptors are called adrenergic receptors. Dopamine is implicated in Parkinson's disease. When there's not enough dopamine, it can cause Parkinson's disease due to the breakdown of the substantia nigra in the nervous system. And serotonin, it's usually known as our happy neurotransmitter. So when you have depression, usually it's low levels of serotonin, and one of the drugs that they typically prescribe for this is SSRIs, selective serotonin reuptake inhibitors. So the three of these neurotransmitters are known as biogenic amines. One neurotransmitter that acts as one neurotransmitter that's an amino acid is GABA. It's an inhibitory neurotransmitter. There's also neuromodulators. They alter the rate of the neurotransmitter release. These are typically neuropeptides. They have long-term effects and can be released along with neurotransmitters. One really good example or class of neuromodulators are opioids. And three types of those are enkephalins, endorphins, and dynorphins. These are natural pain relievers in your body. We also have dissolved gases that can act as neurotransmitters, which indirectly affect the nervous system. Nitric oxide and carbon monoxide are dissolved gases that act as neurotransmitters. Neurotransmitters can open or close chemically gated ion channels, which causes ionotropic effects. They help to either propagate or inhibit ions from crossing. Some examples are acetylcholine, glutamate, and aspartate. Neurotransmitters can also have metabotropic effects, meaning that they have an effect on metabolism when they act as first and second messengers. Some examples of these are epinephrine, norepinephrine, dopamine, serotonin, histamine, and GABA. Postsynaptic potentials can be excitatory or inhibitory. So when potentials are close to the threshold, they're known as facilitated. Integration can lead to summation of these potentials. When you get a lot of stimuli at one time. That's known as temporal summation. And when you have a lot of stimuli at different locations along an axon, that is known as spatial summation. Presynaptic neurons can also be regulated through inhibition or facilitation. One neurotransmitter that acts as a presynaptic facilitator is serotonin. The strength of action potentials is proportional to the frequency of action potentials. So the greater the depolarization, the stronger the impulse, the higher the frequency of action potentials. However, the maximum rate of action potentials will be reached once the relative refractory period is eliminated.